Hi everybody, welcome to our series of podcasts on transport, otherwise known as movement across a membrane. So in this one, the big ideas of what we're going to be looking at are how things are moved across membranes, what happens when things are moved across, how those changes affect the overall life of the cell and how they influence living systems. So let's jump right in, man. So all things being equal, things spread out, right? So we're dealing with here this idea of the second law of thermodynamics, which is the idea of entropy increases as less energy is added into the system. So there's the idea of your bedroom. Okay, so you don't put any energy into it, things get gnarly. Okay, you start putting some energy in, you can keep things organized. So energy gets added to living systems locally. So globally, there is no energy added. So the total net energy isn't added. So the idea is, things spread out and move. Could be one of the explanations as to why the universe is expanding. But nonetheless, things tend to spread out unless we add energy to it, okay, because of that innate nature. So let's start jumping right into the idea of passive transport. So with passive transport, this is what we consider to be the free ride, right? So there's no additional energy needed to be added in. So moving across a semi-permeable membrane, it's the movement of materials from an area of high concentration to low concentration. It's inescapable. If it can pass through a membrane, it's going to happen. Okay, so if a membrane is permeable to water, it's impermeable to the sucrose, water's going to be able to cross, sugar can't. Okay, so water's going to continue to flow across until you end up with the same solute concentration on either side of the membrane. This in turn is going to raise the water levels in that U-tube. Literally a U-tube, not YouTube, U-tube. So when water does this, we call that osmosis. Okay, so the rate of diffusion as it moves, okay, it's the diffusion of water. It's opposite to the direction of the solute because it's going to head from where there's more water to where there's less. Diffusion of water, osmosis. Piece of cake. So here's some idea of when we're looking at diffusion of one solute. So if I have water being able to move across, so we can see that there's a net direction that water moves. It's going to move across, okay, from left to right. Okay, even though individual molecules are bonking around, okay, and moving randomly on their own, there's an overall net diffusion, net direction until equilibrium is reached. And then things are just kind of basically going back and forth, right? So molecules, let's say, of a dye, okay, so they're moving across. Cool. When we have two solutes, you can have two different net diffusions because diffusion is about moving of one solute from one area of where it's highly concentrated to the other. So in the case of the orange ones, they're moving and diffusing net movement from left to right. The purples, though, because there was more started on the right, they're moving from right to left. Okay, so it's because of that internal movement of molecules that they have, that innate movement of molecules, it's inescapable. Okay, it's a property of large concentrations of constantly moving molecules. It's just the way things work. Okay. So we have to think about chemistry when we consider this, okay, because only certain things can cross the bilayer, okay? So what can diffuse directly through the lipid bilayer itself, okay? Not too many things can actually go directly through the bilayer. What does all the other stuff do that can't go through the bilayer? How does it get across? So the answer is if it can't cross directly through the lipid bilayer, it's going to go through transport proteins. This is where we have aquaporins and ion channels. Your aquaporin, that's for water. Water moves through these, through pores, right? Those holes, those protein holes, okay? Ion channels for charged particles, okay? Because of the hydrophobic nature of your tails, it can't really slip through real well. Okay, so channels and carrier proteins are going to control diffusion of charged and polar molecules, okay, through the lipid bilayer. Okay, so in some cases we can actually control diffusion because if the protein channel doesn't work, that's how it can go. Okay, so we can look at it through that way. So if we think about ways on how we're going to control diffusion, one of the ways is going to be active transport. Again, that idea of entropy. So additional energy is going to have to be added to move materials from where they're more where there's less concentration to where there's a high concentration. Energy is necessary because the idea of this is that it's going to cancel out what's happening with diffusion, that inescapable idea of diffusion. So we use active transport for this. Here we need a protein, okay? And this is really important because this is where the energy comes in. So that's really the big reason why the protein is necessary, these particular active transport proteins. So what's happening? We add energy. That energy is what's going to configure the protein to grab whatever that molecule is and shove it across from where it's less concentrated to where it's high concentration. Without that energy, you don't get the shift in the change of the protein structure, which is going to grab that material and move it across. So proton pumps are huge for this. We have to always constantly be moving protons across. Think of things like cell respiration, photosynthesis. That's going to require things like ATP for what's going on. So now look at what's happening here. So see if you can figure this out, right? So the idea of the proton pump is to set up the gradient, right? So we pull tons of hydrogen ions across using energy to create a gradient. So now we have a high gradient of hydrogen ions and we can just let diffusion happen of the hydrogen ions, which is then going to help push sucrose and use our transporters across. So there is a little bit of give and take. I got to put a little energy in to let something else happen. That may not necessarily require it. But again, that second part, this sucrose hydrogen ion cotransporter can't happen unless you have that concentration gradient of hydrogen ions set up, and that's where the proton pump comes in, because that's what hydrogen ions are, right? They're just protons. Okay, so ATP fills our energy requirement, okay? So it's the phosphorylation of ATP is what triggers the conformational change of the proton channel. So ATP comes in, right? Triphosphate, bust off one of those phosphate groups, release a little bit of that energy. That is going to shift the change in the protein, which then lets the stuff happen, okay? So that's what you're seeing when we see things like the sodium-potassium pump. So for some of you, this may be a little bit of a review. For others, welcome to the sodium-potassium pump. Our nervous system relies on sodium-potassium pump, just how it works. So we can't have it without. This is how signals get sent down our nervous cells, our neurons. Okay, so let's take a peek here at what the sodium-potassium pump really talks about. So in the extracellular fluid, inside, outside, okay, you can see this, right? So here we have extracellular high sodium, low potassium. Okay, so that's what's here. So we move the sodium across this way. So that's how we draw in and bring the sodium up that's high. Then this same pump gets shifted when it's open to only allow for potassium to come across, okay? And so potassium goes across. So here we got one, right? Here it comes in, boom, sends them across. That's what's going to set the actual gradient, okay? So this is what we need. High sodium on the outside, low potassium on the outside, high potassium inside, low sodium inside, okay? So we need to actively move and pump sodium back across, okay? Once that's open, see this, the shape now is shifted, where now the shape can grab potassium and actively move that across. So that's just how the sodium-potassium pump works. So you can take a second, maybe jot down a quick thing about how this actually, how it works. And then what we'll do is we'll come back to that when we start talking more about nervous impulse down a neuron, and we'll come back to this. So that ATP is really responsible for setting up concentration gradients. That's mostly what a lot of our transport proteins do for that. So with that, we're going to stop this one, and we're going to then from here move into tonicity and talk about tonicities and how they affect movement of materials across. So we'll see you guys later. Have a good night.